ai điều khiển rồi. Không, hai.
The whole class, before the lecture begins, you can access the Moodle and download the new version of the lecture level 5 nucleus. I have already uploaded this slide on the Moodle. And the new version, if some student who accessed from last week, it changed now. So as we discovered in the first lesson, one cell has three names or three important components. First is cell membrane, second is cytoplasm, and third the nucleus that we're going to discover today. And in the next week we will have a midterms. Today we study the nucleus.
Next week luôn. Wow.
Cô đã nhắc từ tuần trước rồi ạ, tuần sau các bạn sẽ có kiểm tra giữa kỳ ạ. You need to revise from the lesson number 1 to this lesson on the nucleus. In the first part of the lesson today, we study about the nucleus, and at the end of the lesson, if you have time, I will give you, we will solve the exercise together. I will show you the lesson, some question for the final exam in 2020. So you can revise a little bit to prepare for your midterm test. So let's discover what is the nucleus. Cells and nerves are in our bodies. If you have a brain, to control all the activities in our bodies. So the cells, they have one organelles that single functions like our brain. This is the nucleus. It contains all the information inherited from the parents and control all the activity inside the cells. In the first class, we will study the structures of the nucleus. So here you can see that all the eukaryote and prokaryote, they do have the genetic material. However, with the eukaryotic cells, they have a largest compartment here we call the nucleus in the center. Whereas in the prokaryote, they just have one reason or one area that contain the genetic materials we call the nucleolide. So to remind you a little bit, eukaryotes mean here is mean true, and carries means nucleus. So true nucleus, eukaryotic cells have nucleus, whereas prokaryotic cells don't have nucleus, but they have one reason we call the nucleolide. So look at this picture. This is a cell from the humans, cervical carcinoma cells. They have, so under the light microscope, you can see the nucleus that easily seen under the light microscope, and you can see human cervical carcinoma cells here. Tế bào ung thư cổ tử cung ạ. So when you see it under microscope, you can see the red part is the cytoplasm, and the big and very big and center here we have the nucleus. I think I need to explain one term here. In the previous lessons, let's explain the nuclei. Nuclei is the plural form of nucleus, and nucleolide here is the plural form of nucleolide. Now, so whenever it's up here like this, you can see the plural and singular form of this structure. So we go to the major compartment of the nucleus. It has first nuclear envelope, like the cell have cell membrane. Second part, nucleoplasm, like the cell have cytoplasm, is the same. Nucleolus, in the lesson number 2 we discovered. Next is nuclear lamina. And the last part I want to introduce is chromatins. Nuclear lamina is not expressed here, but it will be beneath the inner membrane of the cells here, the nuclear lamina. So we go to the first part, nuclear envelope. First, they are double membrane. To more clarify what is double membranes, I will present here what is the cell membrane here. Cell membrane is made of phospholipids bilayers, right? This is the cell membrane. But what is the double membranes of the nucleus? Is a double membrane. Double layer of membrane. double membrane and it's appear in mitochondrial chloroplast and also the nuclear envelope. And the part I mark green here. The part between two membranes is we call the lumen. Or actually they are interior environments. So the outer nuclear membranes contacts with the membrane of the ER, and the lumens of the nuclear envelope is contacts with the lumen of the ER. For example, here we have the nucleus here, the inner membranes, and then the outer membrane. So here is nuclear membrane. And these are the ER. And the ER that stick with the nuclear membranes, you have the rod ER, right? So it should have some ribosome on it. And you can see the lumens is continuously represent the lumens in the photos here. The lumens of the ER is continuous with the lumens of the nuclear membrane. The purple parts here. So the next informations about the nuclear membranes, they contain numbers of NPCs, nucleopore complexes. So like these cell membranes, they have so many integral membranes to form the channels. And for the nuclear membrane, they do also have some complexes like this, and we call the NPC, which allow things go out and in of our nucleus, of the nucleus. So what is the NPCs? Here is the structure of the NPCs. And large molecules are actually transported between the nucleus and cytoplasms. And for the uncharged molecules, which is smaller than 100 Daltons, can pass through the nuclear envelope. And this is, for the previous lesson, it is passive transport, right? Passive transport with the uncharged molecules smaller than 100 Daltons. And with the molecules more than 100, it must go through the NPC here, and this is active transport. Active transport means it takes energies or things that help bring this molecule through this channel. So the diameters of the NPCs is 9 nanometers. So it's allowed to the proteins up to 40 kilodaltons can pass through these channels. So for more than 100 here is active transport, right? Through the NPC. Big thing is we transport through the NPCs. And the nucleopores are here. Nucleopore complexes, NPCs, is made of 3 to 100 nucleopore proteins, and they can be the gates of transportation of so many molecules. You can see here proteins, RNAs, capsids of the virus, the protein cores of the virus, ions. Iron is good there because they are charged, right? And glucose, they are a little big. ATP is big. So they can allow so many things go in and out of the nucleus. And remember, for me, uncharged molecules smaller than 100 Dalton can go through the nuclear membranes. More than 100. It may appear in your exams. Please notice 100 Daltons must go through the NBC. So the thing that transport into and out of the nuclear membranes is selective transport. They are active but also selective. This is one of the experiments. Here is the experiment in the stenose oxide is one model or animals. It's like it has a shape like the frog. And in the nucleus or nucleoplasms of these cells of stenose oxides, they do have numbers of these structures, nucleoplasmin. You can see here, nucleoplasmin. They are the components of 5 smaller structures, and they made a pentamer here. Pentamer is mean something is made of 5 subunits, right? So one subunit is the 33 Dalton. Kilodalton. 33. And together, when they made a pentamer core, it will up to 100 kilodalton. So this means this cannot transport by simple distribution through the nuclear membranes. So it's too large to be filled to the nucleus. So we have a simple test to see that this structure needs specific molecule or not. The first experiment, they inject it to the cytoplasm. This is a pentamer structure here. And yes, it transports into the nucleus. In the second part, we put the core of the pentamer here, and with only one tail, is also transported into the nucleus. The third experiments, when they insert only the tail in the cytoplasms, it will transport into the center of the nucleus. But when they put only the core without the tails into the cytoplasms, nothing transports into the nucleus. Or I did the core so. So they put the core in the nucleus. They stay there inside the nucleus. They cannot transport back into the cytoplasms. For this experiment, they go to one hypothesis that nuclear pore is selective channel. That allows only proteins with the proper information of their nucleosis sequence to enter. So if you want to bring things from the cytoplasm into the nucleus, you need one specific chains of amino acids or chain or something like a key to open these channels, allow you to bring things into the nucleus. So these signals within the protein to get into the nuclear stretch of the amino acid chains, and we call them the NLS, nuclear localization sequence. Whenever your protein or thing that you want to bring in, stick with this sequence. It will bring in, or if you don't have the NRS, nothing will import into the nucleus. What about the export? This is the import. For the export, they're the same. They have another sequence we call the nuclear export sequence, NES. So if it makes you confused, you just remember for me NES, NLS, and NES. So E is for export. So it's go out. It means from nucleus go to the cytoplasm, and LS localization. So it means go in. Remember for me these two structure, these two sequence, okay? And the nuclear export receptors is need to bind to the protein called the NES. And these were discovered in the case study of the growth of the human HIV virus infected cells. In HIV virus, they have one protein, the red protein. Here the red protein, you can see that in the HIV virus, they do have these proteins and they do contain both of sequence that help these proteins bring things in and also out of the cells. So in this case, the red proteins of the HIV virus bring things in of the cell, usually bring RNA into our nucleus. And by reverse transcript, and they use our machine to transcribe so many viral RNAs here. And this red proteins also brings viral virus with something export out of the cells because they do have NES. So by this protein, the virus can invade our nucleus and use our machines and also export everything out of the nucleus. Another experiment concerning the nucleus membranes. The transport of the most RNAs is unidirectional, one way, from nucleus to the cytoplasm. So here, when we inject the RNAs into the cytoplasm, nothing goes in. But when we input the RNA into the nucleus, they will spread out. All of them will spread out. So it's easy to understand because we have DNAs in the center of the nucleus. They synthesize RNAs, and we need to export RNA out of the nucleus, right? Remember for me the word unidirectional, okay? So we go to the second part of the nucleus is the nucleoplasms. They also call the karyoplasm. So kary is nucleus, right? Karyoplasm, and the nucleoplasm or karyoplasm is they similar to cytoplasm. I'll remind you a little bit in the third lesson. Okay, nucleoplasm plus the cytoplasm we have which part? How do we call them? Plus the cytoplasm. We have when together, Okay, so what is the nuclear plasm? They are gelatinous substance within the nuclear envelope. So everything inside the nuclear envelope lie in the jelly form, and they compose of many waters. Dissolves salt, enzymes, and organic molecules. And they also support the nucleus by helping maintain the shape. And also they provide the mediums for the which is materials, so that enzymes, nucleotides, it can be transported through the nucleus. The next part of the nucleus, we have the nucleolus. So if the nucleus is the center of the cells, we have the nucleolus in the centers of the nucleus. And they are dense membrane-like structure within the nucleus. And in the second lesson, we know that it is the size of ribosome synthesis, right? So the gene encode for the RNA, they are present on multi-chromosomes, and they cluster together and they form a nucleolus subcompartment. And here actually they together, which part of them have the gene encode for RNAs, it will integrate here, and they go very dense here, and these make nucleolus. So here they synthesize the rRNAs to make the ribosome unit. What else? Now the second lesson, ribosomes is RNA plus proteins, right? So here in the center of nucleolus, they synthesize the RNAs, and also they need to import the proteins from the cytoplasm back into the nucleus, and they will stabilize ribosomes in the nucleolus. And the way this appears when the cells undergo divisions, and is reformed after the cell division completes. So it's not a structure, so it's just reasons of nucleus that synthesize the ribosomes. So we can tell is which cell type should have more nucleolus. Normally, generally, one cell have a one nucleolus, but which type cell should have more than one nucleolus? If they have more nucleolus, it means they have a more ribosome. It means they want to increase protein synthesis, right? So if the cell that they want to secrete, or think that they want to secrete many of enzyme or protein, they need to have more than one nucleolus. And some cells have up to 4 nucleoli in the cells. The next structures, we have the nuclear lamina. This is located beneath the inner nuclear membranes. This is the nuclear membranes. This is nucleopore. You can see nuclear membranes. And beneath of them, they have a structure we call the nuclear lamina. And the connex is it physically connects by laminas, and this lamina is integrates with the integral membrane proteins. And of course, the important functions of the lamina is physical support, the shape of the nucleus, and it plays a very important role in the envelope assemblies also. So here more information. The proteins complexes that interact with the nuclear laminas crop the nuclear envelope and link the cytoskeleton. In the lesson 2, I will show you today that in eukaryotic cells, they do have a cytoskeleton. Like you have a bond, the cell have a bond of cells, maintain the shape, have the movement of the cells, and here the nuclear lamina inside the nucleus, they will link to the cytoskeletons of the cells by this structure you can see. And in the yeast cell and some other unicellular eukaryotes, it lack of nuclear lamina. We can explain why in the yeast cell and other unicellular eukaryotes, they are lack of nuclear lamina. Why they do not need the nuclear lamina? Pay attention to the size of the yeast cells and unicellular. In the yeast cell, the nucleus have the size around 1 nanometer. And micrometer. And in the biggest nucleus is up to 10 micrometer. So the nucleus of the yeast cells is too small. So it's one possibility of explanation that the yeast don't need the nuclear lamina. They don't need the physical support because they are too small. The next very important part inside the nucleus, we have the chromatins. And the second part, we go more details of the chromatins. So the chromatins, in general, we can define that they consist of DNAs which contain the heritage information and structure for cell growth, development, and reproduction. And the chromatins, they further classified into heterochromatins, which is highly condensed, and they transcriptional in active form. And other parts, we have the euchromatins, which is less dense compound. So you see the path, this is darker here inside the nucleus. This is the heterochromatin part. Heterochromatin, so it means no transcription. Occur in the dense part here. For the lesser, for the lighter part, in the nucleus, you can see that this could be some transcription. Yes, transcription. And And here's X chromosome occupies in distinct regions or territories, and they will prevent the chromosome become tangling together. They not lie on mass or inside the nucleus. They have the specific reasons for them. Here, inside the nucleus. So here's one short answer questions. We have two micrographs, so the nucleoli of two different cell types. You can tell from this picture which of these two cells is transcribing more of these genes, and explain how you can arrive at your answer. Yes, please.
I think that is the B1 B picture, because in this picture the back point is less and is not as dense as in the A picture, so there would be more chromatin.
More what? Heterochromatin.
Less heterochromatin.
So less heterochromatin compared to the A.
Yes.
And therefore,
they would transcribe more.
yes.
than the A.
So thank you for your answers. You have the correct answers. You can see A's here is mostly in the form of condensed. So it means there's more heterochromatins in A's. B is less condensed, much lighter, and therefore there are so many transcriptional process in the B. Transcriptionally active in the B because they lacked heterochromatin. Compared to the A's, okay? So what could be the advantage of the cells having a nucleus? Having a nucleus allows cells to have much more sophisticated regulation of gene expression. Is prosper in prokaryotic cells. You can see the translation and transcriptions of the in the prokaryotic cells here is quite simple, right? It occurs in the cytoplasms, whereas in the eukaryotic cells, because they are defined nucleus and cytoplasm, so you have a transcriptions in the center nucleus, and then they export RNAs into the cytoplasm to have this translation. Okay. Phiên mã ở trong nhân và dịch mã ở ngoài nhân, whereas in prokaryotic cells, phiên mã and dịch mã they happened at the same time. Okay. And also, you see that the nucleus, they have a double membrane, so it protects the DNAs is much better and more efficiencies. And that's why the eukaryotic cell that contains is more DNA than the prokaryotic cells, in some cases 10,000 times greater than in prokaryotic cells. Because they can protect their genetic materials is more efficient compared to prokaryotic cells. The next characteristics about the nucleus, they can vary, depend on the cell types and also the organisms. They range in size from 1 micron to more than 10 micrometer in diameter. Most cells have a single nucleus, but some cells contains multiple nuclei and few cells like nuclei. Here is 1 micron. I have the examples in the last slide. For example, in the yeast cell, they are unicellular organisms, and they have a very small nucleus, and therefore they have no nuclear lamina. It's one explanation why they don't have nuclear laminas. This plays an important role for the physical support. Who can give me some examples? Which cell have a single nucleus? Which cell have multiple nuclei? And which cell have a lack of nuclei? Have you any ideas about the cell that have a single nucleus, multiple nuclei, lack of nuclei, or lack of nucleus? Now again, nuclei is plural form of the nucleus. Yes, we begin again.
The cell that lack of nuclei is red blood cell.
Yes, very popular examples of the cell like a nuclei.
The cell that only had 1 nuclei is yeast.
Not one nucleus.
One
In the singular form. One nucleus. Actually, all cells inside our body has just one nucleus.
Yeah.
It's not a case. And could you please some examples or one example of the cell should have multiple nuclei?
It is in the cell that in the place when it is
duplicate. It means that the cells in dividing state, right?
Yeah.
Actually, for the cell in dividing state, they have the double. They have 2 sets of the chromosome or chromatins, but it's not the complete. It's not the major nucleus. So think about the cell that they want more energies or they want to excrete more things. Which cell in your body needs more energies?
I think it is muscle.
Yes, muscles. They work very hard, right?
Yes.
They tend to have more. First, mitochondria to generate more energies, and second, they need send more information to the cytoplasm to produce and send more information to the mitochondria to produce the energies for the cells, right?
Yes.
Okay, so we thank you very much for the question, and I think I give you some bonus point for your answer. Sorry. can see the muscle cells. They in the form that they're very long like this. So they be matures by the fusions of the precursor cells. So here, so inside the muscle cell, they have so many nucleus like this because they're very long. And our cells, they do have more mitochondria inside them. Because the mitochondria needs more, some genes, some control genes from the nucleus. They need the information, they need the orders from the nucleus. If we have only nucleus in the center of the muscles, it's hard to send all the commands to the mitochondria that's far away from the nucleus. So they need more mitochondria, they need more nuclei inside the muscle cell. This is the muscle cell. This is the muscle cell. Normal case, single nucleus. But what about the red blood cell? The red blood cells is a special case. The precursor of red blood cell, they do have the nucleus, right? And then it disappeared, no more nucleus. And when the nucleus disappears, it will reshape the red blood cell into biconcave shape, right? Biconcave, hình đĩa lõm hai mặt đúng không ạ? When they have a no more nucleus, it's reshaped into the biconcaves, become mature, the mature red blood cell in the biconcaves. So it's more efficient when they want to bring things. They want to bring oxygen and it's more efficiency. The next part is the origins of nucleus. Start because they have double membrane. And the same, we have the endosymbiotic terrace. That is one cell and gap, another cell, and then primary nucleus with double membrane form. The same with two other organelles that have double membranes. We have mitochondria and chloroplast, right? Three types of organelles have a double membrane, so the origin of them is the endosymbiosis. We have the videos about the nucleus, as maybe we've seen it later. One question is maybe I explain it. Initially, how do have nucleus, but they will lose the nucleus on the maturation of them, right? And then they have no nucleus. They can reform into the biconcave disc. And because there are no nucleus, make them more efficient in the functions, okay? And also, because they have no nucleus, they don't consume the oxygen that they bring. So it's more efficiency. We move to the next part. We have the proteins and also the chromosomes. This is the genetic materials inside the nucleus. So what is the chromatins, what is chromosome, what difference between those? You study in high school. Trách nhiệm sắc với nhiễm sắc thể đúng không ạ? How are they different? Actually, the chromosome is
chromatin is one part of the chromosome, so the chromosome have next to the chromatin that is connected to each other.
is the chromatin. Maybe you mix a mistake about the chromatids. The chromosome and chromatin trách nhiệm sắc là những khác gì ạ? Is it the answer here? Chromosome is the condensed, more condensed part of the chromatin, just only like this. Or we say that chromatins is the lower order of DNA organization of the chromosomes. Against information for the bacteria or genetic materials of the bacteria, this is the nucleoid. And in the eukaryotic cells, is the mass of chromatins within the nucleus. And this is the chromatins when they are condensed in the cell division. They just double and they stick into the centromere. Centromere is the donut. We have more information about the subdivision in the next lessons. But here, during the subdivision, they condense in one phases, and they just they synthesize the doubles and they stick together in the centromere. We call the centromeres, the knot in Vietnamese, and they are chromosomes that link a pair of sister chromatids. The girl that you made a mistake here. It's chromatids. So the subunits of chromatins, we have nucleosomes. It's a little bit difficult to understand, but I will go step by step. We studied together in high school, I remember. So nucleosome is approximately 10 nanometer particles. They contain 200 base of DNAs, 2 copies of H2As, H2B, H3, and H4. Together, here, 2 H2A, 2 H3, H4, they form a histone atomic core. Nucleosome is made by histone atomic core and wrap around approximately 147 nucleotides. And here is more details. These atomic cores and the linked DNAs, they make 200 nucleotides. When we digest them by nuclease, we can separate the nucleosome particle here is more or less 10 nanometer, and here is 11 nanometer. And then when we can dissociate this with the high concentration of soil, and we separate the core and the DNA wrap around them. And the core, histone atomic core, they do have a 2H2A, 2H2B, 2H3, and 2H4, which make the histone atomic core, and wrap aroused by 147 nucleotide pair. And one more histone in this structure is a linker histone, H1. The question that appears so many times in the exam, that is these the histone in the histone atomic core, and whenever you see it appear the H1, it means it's cold, and the H1 is just linker histone. They make to push the nucleosome together and pack them into more compact chromatin fiber. So this is a different level of DNA catchings. DNA double helix, just 2 nanometer. They wrap around the histone atomic core to form nucleosomes. Nucleosome connects together by the linker DNA here, makes a bed on the string to form a chromatin. They can pack and wrap so many times to form 30 nanometers of these structures. And this is the amplified picture of this one part of the chromosomes. This chromosome is more condensed, and it makes a diameter of 700 nanometer. And this is one part of the mitotic chromosomes. This is bed on strings under microscope, and this is 30 nanometer chromatin under microscope. Next information about the chromosomes. They have the bending patterns. By certain staining techniques, cause the chromosome have the appearance of a series of striation called the G bands. Actually, G here is stand for GIMSA. Các bạn nghe quen không ạ? Nhuộm diêm sa của high school không học nào? Em sách cải cách rồi nhỉ? Có không quên, có quên mất hả? G-band is diêm sa, nhuộm diêm sa. So when we're staining the chromosomes, by these techniques, it will appear the lighter band and the dark band. The bands are lower GC content is the nucleotides. G is 1 in C cytosine, okay? The bands are lower GC content than the inner bands. And gene is more concentrated in the GC rich in the band. Our next part, we have studied about the telomeres. So in the two ends of the chromatins or chromosomes, they have a two ends. We call the telomere. And the telomeres, they are made of repeating sequence of TTAGGs on the strand there. And in the reverse, in the complementary strands, we have the AATCCC on the other strands. So they can repeat so many times, from 100 to 2,000 times. 1000-time repetition of this sequence makes the telomere. And the roles of them is protect the ends of the chromatins and maintain the length of the chromatins also. So they require for the stability of the chromosomes, and you know that one cell normally can divide just 20 to 70 times. And then with the dying, the senescence process will be happens, and the cell will be dies. Each time with the cell division, the telomeres will be shortened. After cell division, this telomere will be shortened. Do you know what? Do you know why?
Because of the DNA replication that
I think it's mostly because of the DNA replication. So I won't tell the whole, I don't know the whole detail, but after one mother DNA that replicate into two other DNAs, the result of the two DNA are shorter than the mother because and as it lacks of some nucleotides. I think.
I know. And here they started after cell division. But why?
Well, because cell division involves DNA replication, so it's
Why started?
Because of the way that the DNA is arranged up, like the double helix, a strand of double helix is arranged that two strands of DNA are linked, but they are in reverse sequence. Chụp cả hai cái công là cũng hình như hai cái DNA nó ngược nhau ấy, thế nên là cái việc là nó nhân đôi DNA nó sẽ kiểu nó sẽ không đồng đều ở hai cái strand of DNA đấy.
Đúng rồi, không đều. Vì sao nó lại ngắn đi? The voice. Tại sao lại làm sao?
I think that the DNA in the eukaryotic organism is the flat one, not the circular one. So that when the enzyme primase produce a RNA sequence as ý của em là enzyme primase sẽ tạo ra đoạn mồi ở hai đầu, thì cái adenine polymerase sẽ không thể tổng hợp được cái đoạn mồi đấy để bù cho cái nghĩa là để tổng hợp được ADN thì nó phải tạo ra đoạn mồi để cho cái ADN polymerase biết được, để từ đấy nó tổng hợp cái đoạn mồi dựa trên quy tắc bổ sung. Thì tuy nhiên thì đoạn mồi đấy thì cái ADN polymerase đấy nó không thể quay ngược lại để bổ sung cho cái đoạn mồi đấy được. Thì sau khi nhân đôi xong thì đoạn mồi sẽ bị bay ra khỏi ADN và ADN sẽ thiếu đoạn mồi đấy. Thì cái đâu đấy chính là đoạn mồi.
Cũng gần đúng. Nói chung liên quan đến primer khi mà tổng hợp DNA đúng không? Ngoài cái đoạn mồi RNA ra đấy thì các bạn phải nhớ là nó có hai strain đúng không? One living strain is continuous, and one lucky strain we have some small parts is Okazakis, and they need primers to synthesize during the cell of DNA. When the DNA multiplies, so for the primer, the part of the primers, they have no more nucleotides or no more part supplies for the part of this primer here. And each time the cell division, the cell will be shortened. One part is maybe normally is from 20 base to 100 base. 250 base after the cell division. Because of the primers in the DNA's replicates. And to protect the cells during the cell division, during the DNA replicates, we have this nonsense part, it's called the telomeres. It will protect the ends. Each time of the cell division, the telomeres will be shorter. And when they short enough, or when they short that your cell have no longer divides, your cell will be aging or senescence, bị già hóa, and they will be dying. So it means that when you keep your telomeres, keep the length of the telomeres stable forever, it means your cell will live forever, right? But it's good or bad for your cells. If telomere is not shorter, it's good for or bad for your cells. Your cell will never die.
I think it's bad.
Is that?
Yeah.
What happens?
Because cells, I think cells live forever and they do not die, but new cells are being produced, so and your body can only contain a limited amount of cells, so if you have too much cell, you probably get into some trouble.
Get tumors when you don't have new cells. And actually inside our body, we do have the enzyme that prolong these telomeres. It's called the telomerase. And this kind of enzyme is required for the cells to divide so many times. And in healthy cells, they achieve the necessary balance between the limited cell lifespans. The telomerase is they found to be absent in normal human cells. Or they express just more or less 5% in but in cancer cells, 95 to 29% of them will activate. Third, the cells will be cancerous and they are immortal cells. So we have two ends, telomeres in the chromosome to protect two ends of the chromosomes, and the shorter after the cell division. However, there is one enzyme, telomerase. Now, ACE alone. Suffix ACE means enzymes. This is enzyme prolongs the telomeres. And they do appear in our cells. We have the telomerase in the human normal cell is there is absent or activated just very little amount. But in cancer cells, they can activate 95 or 85 to 95%, and it becomes immortalized cell. They can divide so many times they want. This is in cancer cells. So if you want to treat cancers, we can.
We have to deactivate their telomerase.
Yes, if you want to treat cancer, we can deactivate the telomerase. Do you think it's work?
I think it works better than having to kill every cancer cell because they can just divide it right away. But it is, but I'm not so sure.
Do you think any harmful or any risks when we inhibit the telomerase? So it goes right. So it activates high percent in the cancer cells. And if you want to kill cancers, if you want to make them stop divide infinitely, we can block the telomerase in the cancer cells. Do you think there's any harmful, any risks of this process?
I think it does have certain risks because telomerase, although not every human cell has it, but some, like the reproductive cells, are in need of telomerase so to do their function, because when you kiểu cái hình hành hợp thử thì sẽ phân chia rất là nhiều, đấy là có thể sẽ cần cái telomerase đấy để kéo dài cái telomerase đấy. So when you deactivate the telomerase of the body, you deactivate the telomerase of the cancer cells, but also some of the cells that
The image, right?
yes. So yeah.
Okay, so if you inhibit the telomerase, it could lead you impair the fertility of the cell also. Of the cells. Because in the eggs and the sperm cells and also the stem cell, they will lower. In our bodies, they do have the high level of telomerase, so it could be risk, it could impair the fertility of the cells of our bodies. Next information, the discoveries of the telomeres and the telomerase bring the Nobel Prize for these three scientists. So first, Elizabeth Blackburn. She found the end sequence in the chromosome. In the chromosome or tetrahymeres, one organism lives in the waters, and this is result warnings. In the same years, in the recent year, Jacques, the second scientist, he invents the artificial mini chromosome. artificial. Artificial mini chromosomes. And when they introduce them or inject them into the yeast cells, when they put into the yeast cell, it disappears or it's not work. So nothing protects the chromosomes and it damages the artificial chromosomes. But when they put this sequence plus 2 into these artificial mini chromosomes, it's work. So they suggest that the part that found the end sequence, the discoveries in the tetrameres here, protect the mini chromosomes in the yeast cells. And not 3. The third scientists here, graders, CEG, or students of the blackbird, they discovered the telomerase. So telomerase have to prolong the daughter strands of the DNA, so they keep the DNAs conserved after the cell divisions. And that three scientists here received the Nobel Prize because of this discovery. Here's one information of the proteins during the evolutions of the Indian monarchs here. So initially, chromosome is separate in the Chinese monarchs. Is a deer, hươu Trung Quốc và hươu Ấn Độ. So during the evolutionaries, the chromosome of the Indian monarchs is actually the form of the chromosome of Chinese monarchs fused together. So it just fused together, no effects on number of genes. Or animal. This is one case, a very interesting case of evolutions. One new species forms when the others fuse their chromosomes. So here we have one, 2, 6, 18. 23 pairs of chromosomes by humans, and if they're together here, they have only 3 pairs. They are the same deer, but they are new species. For the next, remember for me in this slide, humans, we do have the 23 pairs of chromosomes. We have a 46 proteins or chromosomes. And the karyotypes, is it the chromosome artificially lined up in order, or we call them the pictures of chromosomes in one organism, we call it karyotypes. More information about the chromosomes is the X chromosomes. They guarantee under the global changes. Here's 2 X chromosomes inactive or embedded in X cell during the embryogenesis of the mammals. The dose compensations. It equalize the level expression of the X-linked gene into sex. You can see in humans, or in the girls, we have 2 X chromosomes, but the gene activates in only 1 chromosome. And here is equal to 1X in man. So it's equal between 2 sexes. In the flies, because the gene is expression in both X chromosomes, so in the organism with X, they are double expression. And in worms, they express, but just half of them. Half of X chromosomes expressed, and then this will be equal to 1 X chromosome expressed in the XE gene here. And here is examples of chromosomes in humans here. In the embryo stage, they have this 1 X chromosome for fathers and 1 X chromosome for mothers. So X females contain 2 X chromosomes, and as early stage of embryo's development, one of these 2 chromosomes become condensed into heterochromates in egg cells, and actually they are randomly become heterochromates. So all the descendants of these origin cells have these chromosomes condensed. Like here, in the descendant of this cell, you can see that all the chromosome X for mothers is condensed, whereas in that cell you can see all the chromosome for father, right? So the mammalian females end up in a mixture of mosaics of cells. Breedings. One part, half a cell is one type and half a cell is other type. Cái này cô giải thích một tí, hơi khó hiểu. Không có phần này. Phần này ví dụ như là female mình có, ở nữ mình có. In females we have 2 X chromosome đúng không ạ? Một từ bố và một từ mẹ. Thế thì trong cái giai đoạn sơ khai của việc phát triển phôi ấy, thì nó sẽ tách ra làm các bạn sẽ thấy 2 tế bào. Một tế bào activate nhiễm sắc thể X từ mẹ và một tế bào activate nhiễm sắc thể X từ bố. Và sau đó nó sẽ phát triển song song với nhau, và như vậy thì ở female, ở các bạn nữ thì các bạn sẽ có những cái mô gọi là mosaics, là các mô khảm là có một vài phần mô là có nhiễm sắc thể X activate từ bố và một vài phần mô có nhiễm thể X activate từ mẹ, nó không đồng đều. Nhưng các bạn nhìn thấy ở đây equal, tức là vì mình có 2 X nhưng lúc nào cũng chỉ có một X activate mà thôi, và như thế nó mới equal, nó cách tính trạng nó mới tương đồng với các tính trạng mà có trên nhiễm sắc thể X ở nam giới ạ. Còn ở đây ví dụ như trong loài ruồi ạ, vì nó activate cả 2 nhiễm sắc thể X cho nên trong kiểu gen XY, cái nhiễm sắc thể X này phải double activate, tức là nó phải activate gấp 2 lần so với bình thường. Hoặc là ở loài worms này thì mỗi nhiễm sắc thể X nó activate có một nửa thôi, cho nên để cho cân bằng với nhiễm sắc thể X activate 100% ở loài có kiểu gen XY. Đó, nó gọi là doses on compensations equal the level of expression of X chromosomes. We have 10 minutes of break, And then when we come back with the exercise to revise for your midterm. Okay?
Điêu, chịu bài không? Điêu.
Có nhắc một tí về thi giữa kỳ này nhiều bạn hỏi quá. Thi giữa kỳ các bạn sẽ làm trên máy của nhà trường, các bạn sẽ được chia làm 3 ca và nhớ theo dõi cái danh sách trong Moodle để biết mình thi ca mấy và vào đúng ca. Các bạn thi khác ca thì cô sẽ từ chối quyền thi của các bạn. Các bạn sẽ làm trắc nghiệm trên máy. Trắc nghiệm sẽ giống như cái quiz mà cô cho các bạn làm, có multiple choice này, có match answer với nhau này, có reorder để tạo thành một câu có nghĩa. Nhiều mã để mỗi ca một mã.
Và ấn
mỗi ca hai mã.
và ấn vào next là coi như phải quay lại đúng không?
Các bạn sẽ finish khi mà các bạn ấn như chữ finish attempt là các bạn sẽ dừng, các bạn sẽ nộp bài.
Không, ý nói là em xong câu 1 đi thì
Đúng rồi, bởi vì để tránh gian lận giữa các bạn thì các câu hỏi một đi không trở lại, các bạn sẽ trả lời từng câu một và không thể back lại. Nhưng các bạn cân đối thời gian cho hợp lý.
vâng, em biết rồi.
Nhớ mang thẻ sinh viên hoặc là chứng minh nhân cư đi nhá, để trước mặt để giám thị kiểm tra.
Wow. Trước đây là anh em toán có 35, 2 toán có 3, không phải là lý hóa C. Toán nhé, toán. Mọi người, nào một câu writing một cái. Writing là làm chưa?
So the whole class we continue the lesson, and in the second part we will first revise the lesson about the membrane and transportation through the membranes. And if we have time, I will continue revise for you all the lessons from the lesson number 1 up to number 4. And my teaching assistant will revise for you the lesson about the membrane and transportation.
Hello everyone, I'm your teaching assistant, and now I have some questions for the revision for the last lecture last week. So the first question is about the characteristic of the plasma membrane. And here you must select the proper position for 1 to 4. So you have 1 minute to think, and I will call the volunteer to answer these questions. 30 seconds. So who can answer these questions? Please read all the and
The plasma membrane is selectively permeable, has control over what molecules and ions can enter and leave the cell. The permeability of the membrane is dependent on the organization and characteristics of the membrane lipids, and proteins. In this way, cell membranes help maintain the state of homeostasis within cells so that organisms can stay alive and healthy.
Okay, so your answer is correct. The second question is, we have a figure of phospholipids and the phospholipid layer, and you will choose the correct answer to field A and B. Who can volunteer?
The A is the hydrophilic and E is the hydrophobic. As you three.
Correct. Question 3. You will have the comparison between active transportation and passive transportation, and there are some characteristics of these two transportations, and we will choose the correct answer to field 1 to 4. This question is quite long, and I will give you half of the minute to think. Okay, so everyone who can.
Mà đâu không được gọi đâu.
So I will choose answer report, right? So for active transportation, molecules move against a concentration gradient. Number 2, molecules move along the concentration gradient for passive. Number 3, cellular energy is required. 4 is not. So that's my answer is B.
Correct. Who can answer this question? It's quite easy. So this question is quite easy, so I will call randomly.
Cái này khó hơn Tú trước.
My answer is B. Question is B.
Not enough. Who can?
I think it's A.
Your correct is correct. And in your real exam, you will maybe have to choose more than one answer. So this question gives you a picture about the plasma membrane with its components. We have 4 tests here, and you will have to choose the proper positions. Half a minute to think, and I will call randomly. Okay, so the time is over and we'll call someone in this area. A girl.
Cholesterol, carbohydrate, proteins, and interval.
Some not true. So who can answer this?
I believe A is carbohydrate, B is cholesterol, C is peripheral protein, and D is integral protein.
Do you remember the function of cholesterol? The function of cholesterol in the last time. Who can remember the function of cholesterol in the last time member?
I think the cholesterol in the plasma membrane will stabilize the plasma membrane against nutrition.
And what is the role of carbohydrate?
The carbohydrate is what the cells recognize each other in the body.
So we call the carbohydrates on the cells is the cell marker. The question 7 is quite easy, so I will call randomly now.
My answer is A.
Are you sure?
See. Oh my God.
So yes, the correct answer is B, and A is not the correct answer because it's the type of water. Question is, in this question I will ask about the correct answer and I will ask you to explain the mechanism. So who can answer this? Half a minute to thinking, to think.
Ôi rồi, anh này có phải quay từ
So who can answer and explain this question?
Okay, so my answer is D because I say the I is not only one dimension. There's only go out and it is only one. The next is the simple term because they have two sides but in one dimension. And the C is the antipicture because the two sides are different direction.
Can you explain the mechanism of this transportation? The mechanism. 勾结或事物卷来一下。 Who can explain the mechanism? No one else?
Also, okay, so the unit porter, it transports only one molecule at a time, but the spin porter transports two molecules at the same time in the same direction, and the anti-porter transports two molecules at the same time but in opposite direction.
In the part B, how the light particle can go through the membrane?
I think because it attracts the red particle, so it just kind of go together through the membrane, I think.
So the correct answer is you see that the red particle is go along its gradient. So
yeah, it's well in the A picture, I think that it goes along the gradient, but in the B picture, the red it goes along the gradient, but the black go against it.
So the red particle goes along with gradient, and when it goes from high to low concentration, they will increase the energy to drive the black particles against
the gradient.
Just against the gradient from low to high here. The same explanation will for C part.
For the C part, I think it's they both go along with they both have the gradient from the outer to inner membrane, but the transporter it needs the black molecule to go outside, so it uses energy to push it outside while maintaining the red to go inside.
It's the same for this problem. Question 9. What are the examples of active transformers? Do you need time to think these questions, or I will go randomly? So please pay attention because this question may be reappear in your real exam. I will call. Who can answer this question?
Qua nghe ở vì đấy.
So this, I will answer this question. This correct answer is D. Or
Anh không quan tâm mà anh cút đi với chó.
So this question here will father with AB blood type and mother with over time. So their offspring can have with over time.
I think the answer will be 4 of them is correct.
Too much. I think the answer is A, B, and C. No, A and B. A and B. Yes, correct. So it's the end of the revisions, and now we will come to part of Dr. Hương.
A and B.
Alright, so we start to revise some questions, and this one question that takes part in the final exam in the year 2019 and 20. Alright, the first question, the cell theories tell us that you can choose more than one answer. I will let you 30 seconds to think about this question, and my TA will ask you randomly. B, D, and E. I think your answer is not covered, it's only K, so the cell is only part of the animal or plant, but you know all the living creature on this planet is not only animal and plant. We have 6 kingdoms, and there are so many other types of organisms. Another answer.
I think it's A, B, and C.
Yes, A, B, and C. The D and E, the K is too narrow, right? When you see the word only, is mostly K is not quite correct. Second question. Name another analysis that primary packaging is a real molecule that will be distributed throughout the cell.
B.
Easy one, right?
B.
So I think it's B.
B, Golgi. So whenever you see the word packaging, it means to draw the Golgi bodies in the cells, right? Yes.
This question is quite easy, and who can answer? Very easy.
Where want to relocate inflame in yourself. Usually,
So who can answer?
vì việc không hiểu gì cả mình chọn đi.
His answer is B.
B.
So in the correct answer, there's two
ER.
two sides that the ribosome can locate in the cell. Is not only plant and animal cells. The first is on the right ER, and the second they can freely in the second level. Next questions.
Viết tắt về câu viết tắt nào. Sustain outside the membrane. Outside the membrane
Near membrane.
What?
Hello, hello. Okay, my answer is C, both A and B.
Đây là đi và
C. D, both A. Yeah, D.
So your answer is D.
Yes.
Both intracellular material and intercellular materials. That is substance outside the membrane. Who have any other answers?
My different answer was the extracellular material.
Yes. Something extra is out of the cell. Extra. This means inside, inner.
So
Yes, it's inside, right? It's just classify the terms that we use in biology. Question about the osmosis. Sorry. You do have the answer here because I marked the rows. So the osmosis can cause the cell burst when it's floated in the hypotonic solutions. So hypotonic solutions is môi trường, right? And in hypotonic solutions, this means the concentrations of the compound of the outer environment is smaller than the inner. The cells and the water will come from the extracellular environments, go into the cell will be swell up and burn. Oh. The question, which of the following is found in plants but not in animal cells? Easy ones. I just shoot the easy question, easy to read the question from the 5 exams for you to revise.
Thế thì A cell wall chắc chắn là A chứ chút A cell wall nhưng mà có cell wall đâu A ok có thể nó có thì có viêm thì cũng sẽ
Master is
Từ sau mắt cận ngồi lên trên này em nhé.
no, I can see the what should I say, the topic. My answer is B.
no
Left here.
ca lĩnh chi
Actually, these plant cells, they have their huge, a giant right here in the center of the cells. But we can find the bacteria in animal cells. They quite small. Sometimes they disappear, but we do find the bacteria inside the animal cell. Other students.
cell membrane thì A
Yeah, the cell wall. We can only find the cell wall in the plant cells. We cannot find any animal cell have a cell wall. And other cell have a cell wall. Other cell with cell walls is we can call the name of fungi and prokaryotic cells in the bacteria and archaeobacteria do have a cell wall. And remember for me the composition of this cell wall. Next question.
Well, I think that I would choose E, A&D.
For respirations, there will be some molecules and also reactions happens, but in response, the concentration gradient is more when it's more focused on the diffusion only. For the explanation is for mentioned about the chains of reactions of the chemical compound. It's quite quite easy ones because I just revised in my lesson today. The ribosome is made of.
I think the answer is D.
So quite easy one, guys. The ribosome is made of proteins and RNA. And remember for me that the eukaryotic ribosome is ATS and prokaryotic one is WS. This question appeared in the quizzes game.
I think the domain of the includes protista and hygiene.
Correct is E.
Okay, but these questions, whenever you see archaea or bacteria, don't choose this. You will have the right answers. Next question.
I believe the answer is secondary structure.
Yes, sure. We have a four-level structure of proteins. Whenever you see alpha helix or beta cyst, it's mean secondaries. The sequence of amino acids, the primaries. Secondary terms and look. So many times we have a tetramers. And more than two chains of amino acids, we have a quaternary structure. answer randomly? I'm afraid that the answer is not true, so please revise carefully. Because you have 1 more week for your midterm test.
I think it is little soon.
Yes, for the last zone, they are the package of enzymes and they enable to digest the molecules. Easy ones and it's appears so many times. So the answer is C. Cells, tissues, organs, and other. Yes. This is different levels of living creatures. The basic unit of cells, the stem cells will mass up the tissues, and various type tissue with different function will make up the organs. Some types of organs connect together will make an organ system, and cell or separate organ system and also the different organ will make up the organisms. Any questions? Answers.
B.
Next question.
C này, C chắc chắn rồi. Có thể là E chăng? E, C, một cái nữa. Sẽ tao nhớ câu này ta làm rồi. There were fan đây bacterial fully inflamed. Đây rồi, lecture 3.
Next question. So moving a big particle into our cell.
My answer is A, endocytosis and exocytosis.
First, I want to here. Maybe last questions. It is classified as the difficult question in the exams